Okay. Well, by my clock, it's a minute after 1, so we should probably go ahead and get started so we stay on schedule. It's my great pleasure to welcome all of you to Ben Rivera's thesis defense. I think I recognize most of the little boxes on my screen, but I should probably introduce myself. My name is Mike Jenkins, and I serve as co-advisor for Ben, along with Dr. Rick Milan. And we also have Jordan Marshall with us, who is Ben's third committee member from Purdue Fort Wayne. So kind of our tradition in FNR is that we start out these defenses with a little bit of information about the student as a means of introduction. So I will do that today. Ben comes to us from Lake Oswego, Oregon. I said that right?
Close enough.
Close enough. So Ben is like a lot of people from the Pacific Northwest, young people, in that, as soon as he could, he fled that region to experience the splendor and the grandeur of the Midwest. And he did so by completing his BS in biology at Kalamazoo College. So Ben joined us in the summer of 2019 at Purdue to work on a project looking at the control of Amur honeysuckle. And most of y'all who are from the Midwest or from the Eastern US in general, if you've looked out your window at any kind of forest, you've probably seen Amur honeysuckle. It's a very problematic invasive. And Ben's project was designed to look at the control of Amur honeysuckle, but also to look at the effects that control could have on other components of the plant community. And then under his own initiative, Ben also added a chapter where he moved away from looking at the control of the species to look at some of the inherent characteristics of the species. In particular, he looked at self-pollination within honeysuckle. And that's kind of an interesting concept biologically, but it's also pretty important to invasion biology. And I'm not going to get into that too much because I don't want to take away from Ben's presentation. But I just want to say that I think I speak for Rick as well when I say that it's been really a pleasure working with Ben. And I think he's done remarkably well during what's been a very weird and trying year for all of us. And, just as the world was starting to kind of go crazy and kind of close in on itself, I had the opportunity to go on what was probably the last sanctioned academic trip for Purdue University when Ben and I were part of a field trip I lead that goes down to the Smokies. And like I said, it was a very weird time to be driving across multiple states at that time because, we were kind of together as a small group, but we were also very isolated because probably more so than scientists do by nature, we were trying to avoid other people. So instead of going anywhere while we were down there, we stayed together in a group in our vans. We spent time in this small campsite together. And we spent our days in the backcountry of this large national park, often trying to go to places where we wouldn't encounter other people very much. So some of these less traveled parts of the park. And I really appreciate the chance to go on that trip because I really got to know the students well, and probably better than I would normally on that trip. And I'm very glad I got the chance to get to know Ben better on that trip. And one thing I learned on that trip, we were all so close together and really getting to know each other well, is that Ben snores to the bejesus. But besides that, I also got to recognize in Ben that he is very enthusiastic about what he's doing. And he's very engaged in the world around him. And I think this makes Ben an outstanding young scientist. And I think it's really kind of helped him on his project, and it's going to help him as he moves forward and continues on and does his PhD at the University of California, Davis. So without taking up more time, without further ado, it is my great pleasure to introduce Ben Rivera. Ben?
Thank you so much for the introduction, Mike. Those are some very kind words. I very much so appreciate it. I should be sharing my screen now. Can I get some thumbs up to confirm that you can see what? Okay, great. Okay, hello. And thank you so much for coming to my thesis defense. I really, really appreciate it. Today I'm going to be talking to you about the invasive shrub Amur honeysuckle. But let's first get some background. So Amur honeysuckle, pictured right there, is an invasive shrub, but it is originally from East Asia. It was most noted for its abundant flowering and its bright red berries, which you can see right there on the picture. Lonicera mackie is its Latin name, but I'll refer to it as Amur honeysuckle. It was actually purposely introduced to the United States multiple times, generally for like ornamental value because of the beautiful flowers, as well as for soil stabilization and wildlife refuge. And the government actually encouraged the planting of Amur honeysuckle all the way up and through to the 1980s. However, what seems to continually happen when the government tries to introduce non-native plants, it escaped and became very invasive. Today, we can see that Amur honeysuckle is spread almost entirely throughout the eastern United States forests. Like Mike said, if you've looked outside in the Midwest at this point, you've probably seen Amur honeysuckle. And there's actually even some population in my home state of Oregon. So why is that important? Why is it so dangerous for Amur honeysuckle to be such an invasive? Well, as you can see from this picture, it creates very, very dense thickets when it invades an ecosystem. And this was even just taken in my front yard. This has it excludes the native plants and can cast a lot of shade and can deeply change the ecosystem. And a major part of this is because Amur honeysuckle, along with a lot of plants from East Asia, actually have extended growing seasons relative to native flora. This allows them to fix more carbon than other ones, grow earlier, take more advantage, as well as shading out other plants in the understory, particularly in the early spring when it makes leaves earlier than anyone else. It also potentially has the ability to produce what's called allelopathic chemicals, which are chemicals that inhibit the germination and growth of other species around it. So it might literally be killing native plants. And what all this leads to is understories that look a lot like this, where there's just not much else growing other than honeysuckle. And that's pretty bad because it has some massive effects on the health of our native ecosystems. It lowers the diversity of plants one can find in an ecosystem, which in turn can prevent the regeneration of oak trees. And oak trees have a variety of importance to not only the ecosystem, but to human services. So like if you like bourbon, it's made out of white oaks. So you want more white oaks, and Amur honeysuckle can prevent that. And it can also affect the microbial communities in the soil, which is really the foundation of everything. So this is what I wanted to study in my thesis. And so how do we even begin to understand the dynamics of an invasive shrub? Well, when you're studying an invasive shrub, or really any invasive species, there are kind of three broad questions. The first one is, what makes it invasive? Like, what are the traits? How does it invade an ecosystem? What about it makes it so much more competitive than a native species? Number two is, well, after it invades an ecosystem, how do you try to control it? How do you try to remove it from the landscape? Is it even possible? And number three, after you do this treatment, what happens to the ecosystem? What's the response of the native flora and sometimes fauna? So my thesis is 3 chapters, and I try to make headway into answering parts of each of these 3 questions. My first chapter is all about the reproductive biology of Amur honeysuckle and what about that makes it so much more invasive than other plants. Number 2 is addressing potentially novel adjuvant to try to increase the efficiency of herbicide so you can use less of it. And number 3 is all about what happens to an ecosystem after you treat it with mulching head. So it also addresses question 2 as well as question 3. So let's get into it. Chapter 1, the self-pollination of Amur honeysuckle. There are many, many aspects to what makes a plant invasive. And there are many answers to that question, but here's one from Herbert Baker. He has this nice condensed list of what makes an ideal weed or invasive plant. I'm not going to make you read it all, but what I want to demonstrate here is that Amur honeysuckle checks nearly every single one of these boxes. It is archetypal to what it is to an invasive shrub, really. But what I got curious about is number 5, self-compatibility. So let's take another step back. What is self-compatibility? Well, there are many terms that too often get used interchangeably when they mean distinct things, but they generally refer to the ability of a plant to reproduce without conspecific pollen or pollen from another individual of the same species. I'll mostly use selfing or self-pollination when I'm talking about this. Now, this became associated with invasive plants because this ability to create self-pollinated seeds was an extreme advantage in what's called long-distance dispersal. When one individually randomly moves far away from all of its other potential reproductive mates, it has a species that has the ability to self-pollinate can establish its own colony by itself for itself without the need for another random chance to happen to find the same location. So that's been called Baker's Law, maybe preemptively, but and that is kind of what we're addressing here, where in Amur honeysuckle. So the question is, does Amur honeysuckle self-pollinate? Well, when I was reading the literature, it was a little bit unclear. Woody plants are frequently don't self-pollinate. It's not, they're associated as not having that ability. And some citations about Amur honeysuckle pretty consistently said that it cannot self-pollinate. However, when I examined those citations more closely, they were all personal communications, or they were referencing a paper that didn't actually address the question of self-compatibility at all. And then some recent results actually indicated that, if you could go ahead and mute yourself, I'd really appreciate it. Recent results indicated that Amur honeysuckle can actually self-pollinate. So Goodall and Iller actually covered branches in bags and found that less than 3% of bag flowers produced a berry. Well, that's a very, very low amount. That's more than zero, if you might notice. Another study was examining the parentage of berries on the edge and the interior of a forest using genetic analysis. And they derived, again, very low rates of selfing, but existing rates that also varied between one another. However, both of these studies, while great, were limited. First and foremost, they only examined a small handful of plants, so increasing sample size was of the great importance. And they both downplayed this result and really focused on outcrossing as the primary means. Outcrossing meaning reproducing with others within your own species, and that they were really focused on that. And they never addressed whether or not the seeds that were created through selfing were viable. They didn't test if they would sprout and create new individuals, and they never examined what might be causing the differences between the rates of selfing that they observed. And so basically these were the limitations and the questions that I tried to address. And here's how I did it. I established 9 replicate sites across the Martel Forest and Richard G. Lugar Forestry Farm, both of which located very near Purdue here in West Central Indiana. They were each categorized into one of 3 different types of invasion. There was heavy invasion, which are like the very dense old thickets that you're probably used to seeing. Light invasions, so like new, younger, a little bit more dispersed invasions that are just starting. And what's called sprouting invasions. So these were areas that had previously been invaded, but a land manager had come and cleared them, but they didn't do enough follow-up treatments so that they actually sprouted back from where they were. The reason I chose these 3 types of invasions is because it might help illuminate a potential mechanism that is controlling the rate of selfing. So for example, if all 3 of these types of invasions were to have the same rate of selfing, then that would indicate that that it is a static, immutable aspect of the species. But if it varied, that might suggest a mechanism of control. So at each site where I could, I had 20 individuals, except for at one site where there weren't that many to find, all displaying ample flowering. We measured their basal diameter and their distance to the nearest flowering conspecific in order to get an access of, how much access to other individuals' pollen does the species have? And we marked 2 branches, and just before they emerged from the flowers, we stuck one of those branches into a bag. And here's the wonderful Sarah Rademacher helping me out with my research. And what we did is we actually fastened these to the base of the branch using a non-absorbent cotton. This allowed us to get a tight seal and would prevent any insects, because it's mostly insect pollinated, from entering the bag and spreading pollen. We then double tied it first with a twist tie, and then again with a zip tie, and then we had a beautiful bag. I should note that these bags are actually specifically designed for breeding purposes, so they allow a lot of airflow and sunlight to enter, so it minimizes harm to the branch as it is. After the growing season had completed in the fall, we counted the number of berries and the number of nodes on each branch that we measured. And this is just to get an idea of how much ability there would have been to create berries. We collected every single closed pollinated berry from the bagged branches. And then we collected 10 open pollinated berries from the same bush. We counted the number of seeds in each berry, and then we performed a germination trial using methods established in the literature. And broadly speaking, here's what we found. First and foremost, Amur honeysuckle can self-pollinate. 28% of our closed pollinated branches did produce at least one berry, which is significant. That's a lot of berries. And this is in spite of seeing more damage to the branch than we initially expected. Additionally, these self-pollinated seeds germinated, and they actually germinated at higher rates compared to open pollinated seeds, which was very surprising to say the least. But then the question is, what is influencing these rates? Now, I would love to sit here for a full hour and talk to you about statistics, but I've heard that that's not the most interesting thing to some people. So I'm going to just say that we put them into some models, did a bunch of fancy model selection, and this is what we came up with. And I'll focus on the most interesting results. What predicted the amount of closed pollinated berries on each bush was invasion type, which is what we were talking about earlier. And what we found that heavy invasions actually had produced the most closed pollinated berries, and light invasions produced the least, with sprouting in the middle. When it comes to the number of seeds, we saw the importance of branch type, so bag versus open branches. Open pollinated branches obviously produced much more seeds, but invasion type also played a major role. However, this was different from the number of closed pollinated seed, number of berry production, excuse me. In this, again, please mute yourself if you can. It is very distracting. Okay, I'm gonna, hold on one second.
I'll just say that
Mute all. Okay, there we go. Sorry about that. For the number of seeds at the heavy invasion, again produced the most number of seeds, which had produced the most number of berries. But this time, instead of light invasions producing the fewest, it was in the middle, and sprouting produced the fewest number of seeds, which is interesting that those didn't mirror each other. In the germination trial, we actually found significance in the interaction between branch type and invasion type. And there's a lot going on here, but what I'd want you to focus on is right here. Within the light invasions, the closed pollinated berries actually germinated at the highest rates, higher than any other group, and significantly higher than the heavy invasions. So while heavy invasions produced the most berries and the most seeds, they germinated at the lowest rate, which is very interesting. So, okay, that was a lot of graphs. What's the point? Well, the take home is that Amur honeysuckle can not only self-pollinate, but the self-pollinated seeds are viable. We found about 1 in 80 flowers produced a berry, which is, again, very low. It's about 1.3% of all flowers. But I would remind you that Amur honeysuckle can produce thousands of flowers in a single year. So even if only 1% of those do produce a berry, that's still a lot of berries, and it still has the potential to establish itself. This can have the effect of reducing what's called the lag period, where species first comes to a new ecosystem where it hasn't been before. This usually takes a handful of years. It can be a long range before it starts expanding rapidly. And so by being able for one individual to establish its own population, it can start building a base for when more dispersion events occur. And then there's the question of quantity versus quality at different life stages. So while heavy invasions produced, again, the most amount of seeds and the most amount of berries, they germinated at lower rates. So maybe earlier on in the light invasions, they're really focused on the strategy might shift, where there's a focus on getting seeds that will germinate rather than at heavy invasions, they just want to make a lot of seeds. And it really stresses the importance of self-compatibility because woody plants are typically not thought of. I think this trait often gets overlooked. And so perhaps we should expand what we're looking at in terms of which plants do have this trait and the effect that it can have on species invasion. Okay, now we're moving on to chapter 2. And I'm going to drink a little bit of water because I am talking a lot. Here we are examining the effect of a novel herbicide adjuvant in treating Amur honeysuckle. So we've established many times over that Amur honeysuckle is a very, very invasive plant. So what do we do about it when it's invaded an ecosystem? Well, there are many methods of treatment, and they often get intermixed with one another, but there are generally 4 broad categories. Number 1 is physical removal. You can think about this like pulling weeds from your garden. You literally just take the plant and you get it out of there with hands or with machinery. We're actually going to be talking about one method called mulching heads that fall into this category in chapter 3. There's also what's called biological control, which for invasive shrubs is pretty new. But there's been a lot of potential in the usage of goats to actually clear areas. Number 3 is fire, which is finally gaining more and more popularity among the scientific community as a means to control. Obviously, indigenous peoples were well aware of that for a long time. And finally, there is chemical treatment, aka herbicide usage. While all 4 of these categories are fantastic and have a lot of potential, they also all have their drawbacks. Physical removal can be ludicrously time consuming, especially when you're trying to scale it up to a landscape level. Biological control is relatively new to the science, and so potential non-target effects are relatively unknown. Fire is very efficient, but there's questions as to whether or not it can actually remove invasive shrubs from the landscape, and in some cases it can even promote more invasion. And chemical treatment, well, there's a lot of issues with herbicides that we're going to dedicate this slide to. In this study, we're specifically looking at glyphosate, aka Roundup, which you might be familiar with. It's the most commonly used herbicide in the world, and even though it's considered relatively benign compared to other herbicides, issues with it are still generally applicable to others. First and foremost, and what's been known about for a long time, but still gaining more popularity, is non-target environmental effects. So glyphosate can maybe harm soil microbes, can spread uncontrollably in aquatic ecosystems, and can affect wildlife. And what maybe you've heard about is questions over whether or not this herbicide is a carcinogen. The International Agency for Research on Cancer, IARC, and the EPA disagree. The IARC says it is, particularly because it tested formulations, which when you buy Roundup from the store, it's almost always mixed with other surfactants and adjuvants that try to increase its efficiency. As a result of these, glyphosate has actually been banned in parts of the EU. Whether or not glyphosate is a carcinogen or having these non-target effects, landowners are particularly concerned with the potential non-target effects of herbicides and specifically of glyphosate. And because the removal of invasive species absolutely depends on the cooperation with landowners, reducing the amount of herbicide used is of the utmost importance. And we have a potential way that we're trying to test of how to do this, and that's by mixing it with 2XL. We're suggesting that 2XL could be an herbicide adjuvant, and what that means is that it could be an additive that increases the efficacy of another element. So in this case, glyphosate. It's not meant to directly kill the plant, it's meant to make glyphosate more efficient so that you can use less of it. 2XL is actually a cocktail of cellulases derived from fungi, particularly Aspergillus niger. And what a cellulase is an enzyme that eats away at the primary components of a plant cell wall. The concept behind using this as an adjuvant is that the 2XL, when applied to the cut stump method of treatment, will eat away at the stump and allow for easier passive diffusion into the vascular tissue of the plant. This will, when it's an easier method of diffusion for the glyphosate, you can use less of it to achieve the same amount of control. Here's how I tested it. I had 4 replicate sites at the Luger farm, which is one of the same sites as before. We had 150 individual Amur honeysuckles per replicate, and they were randomly assigned to one of 15 combinations of glyphosate and 2XL. Glyphosate ranged from 0 to 240 grams per liter, and 2XL ranged from 0 to 50% by volume. We measured the basal diameter of each stem and calculated the total basal diameter of each individual. And we did what's called the cut stump method of treatment that I'm going to show you right now. The cut stump method of treatment is very, very common for invasive shrub removal. It involves that horizontal cut on the individual and then the application of herbicide on top of it, which you can see me doing right here. After we did this, we measured the number of sprouts. You can see one sprout right there, and the length of the longest sprout. And these were measured twice, once in the first year, just a couple of months later, and once in the following year, nearly a full year later. And here's what we found. Again, I would love to talk to you about my models, but that's not why you're here. You're here because of the effect that 2XL might be having. So what we see here is where we applied 0 glyphosate, and we see what we would expect. A lot of sprouting back. These sprouts are long, and it is in both the same and the following year. Excuse me. Now, what we would have expected to see is that as we increased the amount of glyphosate we applied, we would see a gradual shift. We're getting more and more control overall. And then within each level of glyphosate, we would see a decrease as we increase the amount of enzymes, right? So that at this lower level of glyphosate, with a lot of 2XL, we would see less sprouting compared to a higher level of glyphosate with no 2XL. That's what we were hoping to see. What we saw was different in that we just had low amount of sprouting across any concentration of glyphosate. Additionally, just for reference, there were no real differences between the lengths of the longest sprouts. Now, what does this result mean? Well, the primary goal of our study was to reduce the amount of herbicide necessary for the control of Amur honeysuckle. Now, typical recommendations for glyphosate are between 98 and 122 grams per liter, which we found similar levels of control with applications at that amount compared to lower dosages. So we already see that we can apply nearly a third of what is recommended and see the same amount of control, which is really interesting. And what it suggests is that it might be time to reevaluate those recommendations for Amur honeysuckle control and maybe for other species. However, what we found is that 2XL did not help as an herbicide adjuvant. But why it didn't help is actually very interesting. What we were assuming is that glyphosate uses passive diffusion to enter the vascular tissue. However, because 2XL was harming the cell walls, it could actually be using active transportation. So that when the cell wall got damaged, that actually decreased glyphosate's ability to be taken up by the plant, which if you see on this previous slide, there was actually a slight increase in the number of sprouts at this low density, which is not at all what we expected. It looks like 2XL made glyphosate worse. So perhaps this mechanism of uptake is not well described, but there is some more evidence that it requires active transportation. So maybe an intact cell wall is a mechanism that we can actually exploit and find an adjuvant later on that will help glyphosate enter the vascular tissue more efficiently. All right, now we're on to my final chapter, chapter 3. This is all about the mulching head. Now, this is actually a continuation of a previous study. You can see Graham Frank, Mike's previous master student. And his motivations were actually very similar to mine. You want to control Amur honeysuckle more effectively and maybe reduce herbicide usage along the way. And the way that he suggested this be done is through what's called a mulching head. There are a lot of names for this. It could be a rotary mulching head, a forestry mulching head, wood shredders, mastication head. I'm going to stick with mulching head. And you can see that right here in the bottom right. It's basically a big grinder that can just grind up any and everything in front of it. He tested it and compared it to the cut stump method of control, which I had shown previously in my last chapter. And he actually found that there were similar rates of control between the two. But this is an herbicide-free alternative, which is pretty impressive. The concept behind it is that the mulching head, as it digs down, and we'll see a video of it later on, can shatter the root collar or the base of the Amur honeysuckle. And while some woody plant species have the ability to make new sprouts somewhere else along its root system, it would appear, at least in our observations, that Amur honeysuckle does not have that ability and can only sprout from the base. So if you destroy that base, then you can control the individual. However, a question was left remaining in that how does the intensity or how far it digs down into the soil affect this control and the ecosystem? And Graham Frank started to examine that. He established 3 replicate sites at the Luger Farm and the Purdue Wildlife Area, which is not far from Martell Forest. At each site, there were 8. Oh, this also included an untreated reference site, which is just all the Amur honeysuckle was just left intact. Obviously, all these sites were heavily invaded with Amur honeysuckle. At each one, there were 8 plots. Before any treatment, Graham Frank measured the percent cover of plant species in their herbaceous layer in 1 meter quadrats in each of the cardinal directions around the center. And then he treated them with the mulching head in just the same way that he had in his other study. But this time, he recorded the intensity, or again, how far down into the soil the treatment was digging, using wooden stakes that were pre-marked. So where they shattered along their base is how far down, and they're averaged for each plot. After treatment, he once again measured the volume of the stems sprouting from the stumps, but this was just in the same year as treatment as he graduated with his master's. I would come back multiple years later and remeasure the same sites for the sprouting volume as well as their herbaceous layer percent cover. Now, to see what the mulching head looks like, you can see that it's this big, heavy piece of machinery that is just gobbling up every single thing in front of it. And by intensity, we mean how far that arm goes down. So it's measured in centimeters below the soil surface. And what Graham Frank found in the same year as treatment was a very nice negative relationship. So as intensity increased, as you dug deeper into the soil surface, we actually found less sprouting back of Amur honeysuckle. So what that means is you shattered more root collars and prevented more sprouting, which led to less Amur honeysuckle. This is just within the same year. So some more questions arose from this. And these are the questions that found my study. First and foremost is, does this trend hold over multiple years? Then how does the herbaceous plant community respond to these deeper intensities? And is there a trade-off where the digger you deep down, the more you control, but the less good it is for the herbaceous community? Well, I addressed these in order, and first and foremost, over 3 years later, we found the same negative relationship. As one digs deeper and deeper into the soil surface, Amur honeysuckle remains less. There's less volume of Amur honeysuckle up to at least 3 years later, according to our study. And that's already great because consistency is very important to land managers. If they can apply this treatment and know how it's going to work, then they are feeling pretty good. But the question remains, how does this affect the herbaceous plant community? Well, again, I really would love to talk to you about statistics, and there's so much more I did in this project, but I'm going to try to condense it as much as I can for y'all. I used what's called a canonical correspondence analysis, also known as a CCA. And it's a multivariate approach. And basically, like the linear regression we just saw, it takes multiple X's and multiple Y's. And it explains the Y's by using the X's. That might sound a little bit confusing, and I'm sorry. But essentially what it means is that we took all the pre-treatment data, including like plant diversity and the intensity of treatment and which site it was at, and we used that to explain what the sites look like in some of the similar variables, plant community, but also Amur honeysuckle presence in the post-treatment slides. What you need to know is that the red dotted arrows you're about to see are going to explain the solid blue arrows. Red is pre-treatment, blue is post-treatment. And so here's what we found. First and foremost, this is a lot, I know. We're going to hone in a second, but what's important to note is that we found significance in this model. So pre-treatment data that we had did significantly explain post-treatment results. And what we really want to understand is the role of intensity. So I'm going to zoom in there. Because the explanatory variable of intensity is trending in the same direction as these blue arrows, that means that intensity is explaining these. Now, what variables are these? Well, I'm glad you asked. They are mostly plant diversity increases with increased intensity, as well as plant richness, plant evenness, and C average, as well as FQI. Now, FQI and C average are measures of the value to conservation dictated by the state. So it's different from state to state, but it's essentially, are the plants there something that is somewhat subjectively defined as being contributing to the conservation value of an area. So the point that this is making is that with more intensity, you're getting more plant diversity and higher amounts of conservation value in your resulting plots, which is kind of the opposite of what we expected. However, we should note that the average C value, again, conservation value, for all our post-treatment plots was only 2.73 out of a possible 10. So these are very, very low numbers, and that becomes important in just a bit. Again, I showed you a lot of graphs, and I know graphs are scary, but let's bring it back to the original questions that we had. And now I have some answers. So does the trend hold over multiple years? Yes, it would seem that deeper intensities can control Amur honeysuckle volume over at least up to 3 years, which is very important to land managers. The herbaceous plant community responded by increases in diversity and conservation value as you increased the soil disturbance from increased the intensity of treatment. And is there this trade-off that we were expecting to see between increased control and herbaceous plant community response? We didn't see one here. Now, what does this mean? It means that mulching head treatment is showing some strong promise as a control technique for Amur honeysuckle. It is able to, deeper and deeper intensities even seem to be a pretty positive thing because it controls sprouting and promotes diversity. However, I should note, and you'll see here in my limitations, that our sites were historically very disturbed and very previously invaded. So we had low conservation value across the board. So with more soil disturbance, we didn't see a negative effect on native plant species because there weren't really that many native plant species to begin with. So while mulching head treatment can be expensive because you have to rent this big equipment, it can be scalable if you're looking at doing a lot of land, which is really great. And especially because it can either replace herbicide treatments or you can use it and then follow-up treatments with deeper intensities will require less herbicide treatment later on. I would also note that this study that we did tried to avoid damage to native species and minimize soil disturbance. So the depth at which we saw as a maximum was only 1.5 centimeters below the soil surface. So in other situations where these precautions aren't taken, you might see even more soil disturbance, which could affect things. particularly in sites where there are a lot of native species to try and preserve. So examining in other forest types and at even deeper intensities might be required to fully understand the implications of this treatment. So in conclusion, what did my thesis contribute? I'm obviously very proud of the work I've done, and I think one of the things I'm most proud of is that it contributed both like immediate pragmatic insights as well as kind of some conceptual ideas into Amur honeysuckle as an invasive species and the control of Amur honeysuckle. Pragmatically and most immediately, what I can say is that my research has suggested that we can lower the amount of herbicide necessary to control Amur honeysuckle invasions, at least in some cases. Less glyphosate in our cut stump method was achieving the same level of control as higher levels of glyphosate. And the mulching head study showed an alternative in initial treatment that also promoted more diverse and more valuable and herbaceous communities more valuable to conservation. Conceptually, we added a little bit more evidence that glyphosate's mode of transportation into the vascular tissue of Amur honeysuckle is requiring active transportation, which again, if we can understand that mechanism better, we can maybe find other adjuvants that can even further increase the efficacy of glyphosate. And finally, Amur honeysuckle is self-compatible. It can self-pollinate and establish viable seeds all by itself. Thank you so much for coming. I really appreciate any and everyone who showed up. I could give so many thanks to so many people. Y'all are amazing. Of course, my wonderful friends and family, and my great and very, very patient advisors, Mike and Rick. Jim McKenna, you're always there to talk things through with me. And I have to give a very heartfelt shout out to Sarah Rademacher for being one of the sole reasons I was able to stay sane over the last 2 years. So thank you so much. And I think here are some references. I think I have time for questions.
So you can either raise your hand physically or digitally, and Ben is hosting this meeting, so I'll let him recognize people and respond to their questions.
Well, I'll ask a question. First of all, great, really great presentation, super clear explanation, and really cool to see some impactful work. My kind of question was, one of the things that I thought was really notable is that you were getting these, really high rates of berry production. And, like in those other studies as well, they're like, well, it's only 3%, but it seems like what the numbers, the estimates you're getting just by the requirements of the experiment are like the extreme low end, right? I mean, if it weren't restricted to just one branch and you weren't restricting the insects, it could be way, way higher, I would think.
Yes, I absolutely agree. One of the issues we were facing with the self-pollination study is that we actually saw more branch death than we expected with the bags. And this is due to like the very last heat wave of the summer that year really just knocked out a bunch of our branches, which was just a real bummer because that could have led to even more berry development and more seeds. And so definitely, I think we're on the very low end of what Amur honeysuckle's self-compatibility potential, and it's a potential to produce self-pollinated berries when excluded from other pollen.
That's cool. Thank you.
Yeah.
Related to that same study, I don't think you really define the difference between heavy and light. And so I've done work where trying to define heavy and light invasions for whatever we're trying to do is really difficult, especially across later sites. So how did you define heavy and light classifications for your sites, but then also how does that then translate to applying this to other locations?
So great question. one thing I didn't have the time or space to show was that we actually double checked to make sure that there were differences between these communities. So our first evaluation was subjective. It can be pretty easy to, when you're walking out there, to smack your head into a heavy invasion. Those are very easy to define. The light invasions, we had to search more because they were smaller plants. So we had to consult with Brian Beheeler, who was, again, another person I really should have shouted out for all of his help. But we were able to find some light invasions, and we, after we measured distance to nearest conspecific and basal diameter, there were significant differences between these two groups in both of those factors. So heavy invasions were larger individuals and closer together compared to light invasions, which were smaller and further apart.
But I guess my question is kind of also, if you tried to apply this to another location, couldn't you also find differences in distance and basal diameter, even if they would technically have come under your light definition for your study? And if you're in another state, I mean, you go to Colorado, where maybe they have incipient populations establishing and they don't really have that difference, couldn't they still find a difference in what would fall under your light definition, but still be differences in distance and diameter? So I guess my question is more of, how do you take what you observed and what you categorized and make it into a threshold that can be used at a different location?
Yeah, so obviously comparisons between different forest sites would be in different states would be of the utmost importance. But I think what we're under the assumption here is that at our heavy sites, we're kind of at the saturation point of Amur honeysuckle invasion, where it's as much as you can get in that understory is kind of already there. And we're lucky that these sites are some of the oldest invaded sites anywhere in the United States. So it's very likely that these sites are at that saturation point. So we can kind of use that, which of course will vary by site, but say that we're at this saturation point, and that might be at least somewhat generally applicable to other forest types. Any other questions?
Yeah, Ben, I really enjoyed your presentation. Nice work. And
Thank you.
I was interested in your third chapter where, you used this mulching head to rogue out honeysuckle and looked at the diversity of the plant communities that come up underneath. It's like a desert underneath these things. Right?
It truly is, yeah.
Now, I just wondered what you know about those communities, and if you saw any kind of regeneration of things from the seed bed coming out, or, if you characterize what was part of these communities, and if you saw.
You mean post-treatment, right?
Yeah, post-treatment.
Yeah. So one thing, my biggest takeaway from those post-treatment communities is that there wasn't a bunch of, there really wasn't a strong seed bank ahead of time, which is why we saw such low C values and FQIs. Yeah. We saw a bunch of goldenrod and a bunch of clearweed, which here is, it's pretty much everywhere already. Native, but not something that land managers are particularly conscious about approaching, which is why I would suggest that trying experimenting with this in forest sites that maybe have some species already established that are of particular interest to land managers might be a really good idea before broadly applying it.
Thanks.
I can actually show you. Hold on. I snuck this in there at the very end. I can show you which species were driving differences between different groups of intensity. And if you can see that, most of the contributing species here are again, clearweed and and I know, which was surprising.
From Island, you stuck that in there.
Yeah. So, but a lot of
You'll get yours, Ginzel.
So that's so right. It's not a bunch of species that are of particular interest driving differences between intensities.
Thanks.
Ben, this is Lenny Farley. I'm an extension forester in the department, and I want to congratulate you. Great work.
Thank you.
I think there's going to be some real strong interest in these results because the practitioners are always looking for ways to apply less herbicide.
Right.
So would you make some suggestions about maybe follow-up experiments that we need to do to really set the standard for how much glyphosate is enough and how much is not enough?
Yeah. Oh, thank you so much for the question. Obviously, I mean, examining ways to lower glyphosate usage is a big motivation behind my entire thesis. I would say that first and foremost, it's worth examining concentrations lower than 30 grams per liter, which is already very low. Again, that's like less than a third or like about a fourth of what is often recommended. So, but examining like 0 to 30 in 5 grams per liter increments might be worthwhile. And examining those not only in 1 year, but over multiple years would be my first instinct. And it's possible, maybe even worth examining 2XL again at these even lower concentrations, where maybe a difference might pop up, or at least the 2XL's inhibition of glyphosate might become even more apparent at even lower concentrations. But I think the cut stump method is very efficient, and it's a nice balance between physical removal and treatment with herbicide. So I think that's a treatment I would stick with and kind of continually go down the ladder until you can find the least amount of herbicide you can apply.
Well, I don't want to cut the discussion off because there have been a lot of great questions, but I also don't want to delay the closed door session either. So if there aren't any more questions, why don't we give Ben another virtual round of applause. Thank you all for participating. This has been, there are more participants than I expected, not just because it's a Zoom meeting, but also because it's during the summer and a lot of people are away. So thank you all for participating. It was a great turnout. And thanks again, Ben, you did a wonderful job.
Thank you.